everyone! Today we are going to be extracting DNA from strawberries. As we all know, DNA is the genetic blueprint for all living things, including strawberries. We are using strawberries specifically because strawberries are octoploid, so they have 8 copies of each chromosome, meaning they contain more DNA than most organisms, which as a result produces a lot of visible DNA. Before we start, here's what we'll need. Around 4 to 5 fresh strawberries, 1 teaspoon salt, 1 tablespoon dish soap, half a cup warm water, 1 Ziploc bag, rubbing alcohol, and a cheesecloth or a coffee filter. As we go through each step of the DNA extraction process, the strawberry undergoes several changes at the cellular level. First, we mash 4 to 5 strawberries inside a Ziploc bag. The strawberry should turn into a soupy red mush. By doing this step, we are physically breaking apart the fruit's cell walls. This gives us better access to the DNA. Next, in a cup, we are going to mix half cup water, 1 tablespoon dish soap, and 1 teaspoon of salt with the strawberry mush and stir gently. Soap helps dissolve the cell membrane and nuclear membrane of the strawberries as they are made of fats. The strawberry mixture gets foamier and slimy, and the liquid part turns more transparent because the soap is dissolving fats. Breaking the cell membrane. Dish soap contains detergents that can dissolve fats. The cell membrane and nuclear membrane are made of lipid bilayers. The soap molecules surround and break apart these fats, disrupting the membranes. Salt helps by pulling water out of the cells, making them burst open. It also helps DNA clump together once it's released by neutralizing their negative charge. Step 3 is filtering the mixture to separate the DNA from the solid parts, or the cell debris. The coffee filter or cheesecloth separates the big strawberry chunks from the dissolved DNA solution. This step removes plant debris like cell walls and proteins, leaving behind a clear liquid that contains the DNA. In step 4, we add the cold alcohol for the DNA to precipitate out. DNA is soluble in water, but not in alcohol, so when you add cold rubbing alcohol, DNA clumps together and floats to the top. The cold temperature helps slow down molecule movement, making the DNA separate faster. A white stringy substance will appear at the surface, which floats like spider webs. The alcohol and water stay separated into layers. What we see as a result after breaking the cell walls, dissolving the cell membranes, and filtering out the debris are isolated DNA strands that we have made visible.